The overall goal of the following experiment is to discover and characterize novel gene function using gene annotation via combined analysis of metabolite profiling and co-expression analysis. This is achieved by consideration of experimental design with a focus on particular metabolic pathways and plant species. As a second step, analytical approaches are used to construct the whole metabolic pathway based on the chemical structures of detected compounds. Next, the structure of the predicted pathway is integrated with co-expression analysis for narrowing down candidate genes which putatively indicate key genes of metabolism. Ultimately, results are obtained by performing minimum sets of experiments with efficient use of several bioresources in order to characterize functional identification of genes.
We first had the idea for this method when we found the very high correlation of Arabidopsis flavonoid biosynthetic genes in the co-expression database named ATET. We considered that this strategy is one of the fastest ways to find the novel gene functions with in silico data's publicly available database and efficient use of bioresources.
To begin the extraction, harvest the plant materials and freeze immediately in liquid nitrogen. Once frozen, powder the plant materials using a mixer mill. Aliquot the frozen plant material into a 2 ml Eppendorf tube, and then add a metal ball to the tube. Add 250 microliters of extraction buffer per 50 milligrams of sample. The extraction buffer contains the internal standard for